All right, good morning, everybody. I shall start with the obligatory sleepy puppy shots. There's one. And there she is. There's the other one. It's a hard life being a dog. Yeah, exactly. Get fed whatever you want. Well, not whatever you want, but most what you want. Go for walks, sleep on the couch. Sounds pretty awesome. Okay, so today we're going to be starting Mendelian genetics, or really the details of transmission genetics, right? Genetics at the level of kind of individuals from one generation to the other. All right, so there's a whole bunch of language associated with this topic, which we'll start off in a little bit. And really, this is about math. Don't you start barking. Right? A lot of it's really just probability, right? And so there are some very kind of codified ways of thinking about this, which are really the monohybrid and dihybrid crosses that Mendel described. But really, it's just a way of thinking, like thought exercises in a sense, or a framework to work on, right? And so the key to really getting Mendelian inheritance, Mendelian genetics, is understanding those very specific ways of looking at transmission genetics, right? They're very specific. They do not change, right? So once you've learned them, you're good, right? There's nothing, well, not that there's nothing else that you need to learn, but once you've got them in your head, you're pretty much set. So I'm going to start off with just some kind of definitions. It's a little tedious, I know, but they're important because without them, you won't understand what I'm talking about, right? And then some of them you know, some of them that you may think you know, and some of them you may not know, right? And so really when we're talking about Mendelian genetics, there's a few things that we care about, what we're really talking about. Those are the genotype and the phenotype, really. The genotype being the underlying cause of the phenotype that you observe. Or if you look at it the other way around, the phenotype is the expression of the genotype. So you can kind of look at it two different ways. Now, you'll not always be able to see what the genotype of that individual is from its phenotype. We'll get to why, in a bit. So genotype goes to phenotype. Phenotype does not necessarily lead back to genotype. It's kind of like the sequence hypothesis and central dogma in a way. That's something that's really important to remember. When we're talking about genotype, we're talking about what alleles an individual has for a particular gene. So alleles are different forms of the gene. They don't have to be functionally different. They can simply be a single base change, right? Even if that doesn't change the amino acid sequence. But when we're talking about alleles in the sense of Mendelian inheritance, they do have a functional difference. Right, so don't always assume that an allele actually changes anything. In the context of what we're talking about, it does. And locus is really where that gene is on a chromosome. When you're talking about just one gene, it doesn't really make any difference where it is, to be honest, right? In terms of like a monohybrid cross. When you start talking about 2 or more genes, now it starts to have an impact, right? Our assumption in Mendelian genetics is that those genes are on different chromosomes. That's one of the assumptions, and we'll get to assumptions in a little bit. All right, so that's kind of basic stuff. The big deal here is the allele, right? Being a different form of the same gene. And that different form essentially being a different sequence, right? Most of it's the same, but there will be a change somewhere which changes its function in the context of what we're looking at. This is all pretty straightforward too, homozygous, heterozygous. Homozygous, so we're always talking about diploid organisms, which we are, right? So every diploid organism has 2 copies of a gene. They may be the same, in which case it'll be homozygous. They might be different, in which case it'll be heterozygous, right? And when we talk about alleles in the context of this material, we talk about dominant versus recessive, right? So a recessive allele is only expressed when it's homozygous, right? Or a recessive trait is only expressed when you have 2 recessive alleles. Now, dominant trait, on the other hand, is expressed when you only have 1 dominant allele. You only need 1, and typically when we talk about dominance, we're talking about something that functions. Another way of calling this is a wild type, right? So wild type allele is really what we mean these are all pretty much the same. Now, there are other kinds of dominant allele, but we're not going to be getting into those here. You can have things that are they're called dominant negative, but yeah, those are kind of tricky to explain. Right, that's something that hopefully you'll come across as you get into, molecular biology or something later on in your degree. And so the fact that you can have a dominant or functioning trait, a wild type phenotype, is actually, sorry, I got to change something here. It just occurred to me. This is something that has I keep forgetting to change. I'll upload a new version after class. And so the reason why I'm changing this, thanks Bridget for posting that. Would you like me to post that in an announcement? Just for anybody that isn't present today? Okay, I'll make myself a note so I don't forget. Cool. Oh, and before I forget too, I can't remember if it was in this class or my other section, but someone asked about getting the audio walkthroughs, the audio recordings transcribed into text. And I looked into it myself, and it's crazy expensive. But I'm talking with DSS, who have that software, to see whether they'll be able to do it for me or not. I haven't come to any kind of outcome yet, but it's something that I'm looking into. So I can't remember if it was someone in this class or the other one, but either way, if I get that done, then I'll let you all know. Okay, so one reason why I had a little pedantic tick about that is the trait is the thing that you're looking at. Right, and so this is an important distinction, right? Because it's quite easy to get tied in knots when we're talking about these sort of things. And sometimes I catch myself out too. So, for example, hair color. Right, we're not saying what hair color you have, we're just saying we're looking at your hair color. That's the trait that we're investigating or talking about. And the phenotype, what hair color you have. Right? And then the genotype is obviously the collection of alleles for different genes which are responsible for that particular phenotype. That's probably worth writing down too. So you can see it's kind of important to get the language right, because we're talking about something very specific here. And so we have to talk about it carefully. Otherwise, we'll end up potentially giving the wrong information or coming to the wrong conclusion. Let me just double check. I think this is, oh yeah, we have next Tuesday too. So I can kind of take my time and go steady. So yeah, lots of, I always tell this to my classes. I don't think I've told you yet. But when I lived in Colombia in South America, I didn't know how to speak Spanish. So I'm actually kind of giving you a bit of a like inside information that I can sort of speak Spanish, not super well. And before going, I couldn't speak any Spanish. And so my uncle said, oh, you just add O or A to the end of any Spanish word. And there you go, you're speaking Spanish. That was his advice. Obviously, as you probably know, it doesn't quite work that way. But one thing that I did, which helped a great deal, is I kept a little book in my pocket, a little kind of notebook, I guess, and just wrote down every word that I come across that day, like every new word, Spanish, and what it meant, because I was working with people who could speak English as well. I was working in a research lab. So every night I just go through that and kind of just try and kind of quiz myself every page. And it got to the point where, you know, the earlier pages, I just didn't, you know, need to look at anymore because I knew those words. So when you're learning a language, and really what we're learning is no different, that kind of approach can really help. Just so that you just kind of get that stuff embedded, so you don't even need to think about what you're saying. You just, it comes naturally, essentially. So that may help for some people too. Okay, now we get into some of the terminology that we're going to be using when we talk about Mendelian genetics specifically, right? And so there are a bunch of assumptions. I'll write those out in a little bit because they're very important. One of them is that the parents are true breeding. Whereas actually that's not an assumption, that's part of the cross. That's a little bit different. And so what we mean by true breeding is pretty much what you think it does, right? If we're talking about, well, for example, this mutt here is about the opposite of true breeding as you can get. We have absolutely no idea what she is. Actually, her siblings, because they're part of a group that were rescued at the same time, one of the people at the rescue center actually got one of them like 23andMe, but for dogs. I don't know if you've heard about that. It's kind of neat to see what breed they were. And it came out with, I think it was like 9 or 10 different potential breeds, including some that I've never heard of. I wouldn't even repeat them. I don't even know how to spell them or say them. And so she is a just complete collection of different alleles for different genes, right? She's about the opposite of true breeding as you can get. Because true breeding literally means if you take two individuals, like two siblings, and breed them, their offspring will be identical to them. And if you do the same again and the same again and the same again, you'll never see any different phenotype. You'll always only ever see the same phenotype. And the reason for that is because those individuals are homozygous. Now, we typically refer to that in terms of 1 or 2 or 3 genes or whatever, but it's the same terminology that dog breeders use. If you cross a Shih Tzu with a Shih Tzu and they're true breeding, you're only going to get Shih Tzus, right? You're not going to get some kind of weird Doberman Shih Tzu like sausage dog popping out. And so if you never see anything different, it's because there are no different alleles present in that individual for that gene. They are homozygous. Doesn't tell you whether they're homozygous dominant or recessive, just that they're homozygous. Because if they're heterozygous, then eventually you'll see something different pop out. Hybrid is essentially a heterozygous individual, right, which has 2 different alleles, which is produced from crossing 2 true breeding or the opposite true breeding varieties. Now, monohybrid and the dihybrid cross really are very formal crosses, right? And I use that word very deliberately. They involve either 1 or 2 genes. Essentially, they have 1 or 2 traits that you're interested in. Monohybrid is 1, dihybrid is 2. Right. These are very formal crosses, and they are always the same. And so the neat thing about that means I can tell you part of the information for a monohybrid or dihybrid, and you can tell me the rest, right? Because they always have the same structure. And that structure is always of 3 generations. The parental generation, where the parents are true breeding, they're homozygous, and opposite. The F1 or filial generation, which are the hybrid offspring from the parents. And then these hybrid or heterozygous offspring are crossed together to produce the F2 generation. And it always happens exactly the same way. Now we have a bunch of assumptions for monohybrid and dihybrid crosses. or really for Mendelian genetics. And just to kind of give you a heads up, when we talk about Mendelian genetics, we're talking about this very formalized, really quite simple genetics, dealing with few genes. Louise, it is, but I'm going to upload a new version of it after class. Actually, make a note of that too, because I changed some of the text in one of the slides. All right. Where was that? Yeah, so something that's quite, painfully obvious is that this doesn't apply to most genetics, right? And so most transmission genetics are non-Mendelian. So there's a whole bunch, actually there's way more that we're going to talk about under the label of non-Mendelian genetics or non-Mendelian transmission, right? And that deals with all of those kind of more real life situations that Mendelian genetics does not cover. So when I keep talking about Mendelian genetics, it's because there's a whole bunch of non-Mendelian genetics afterwards. So the assumptions for Mendelian genetics are: 1, alleles segregate into gametes independently or randomly. And that's called the law of segregation. Right, so if you have, obviously if you have 2 of the same alleles, then those gametes are going to get the same allele. But if you have 2 different alleles, you're heterozygous for a gene, then which gamete gets which allele, the dominant or the recessive, is random. Right, and so if you're dealing with a heterozygote, that means you're going to get 2 gametes or 2 types of gametes in a 50-50 ratio. I hate typing out heterozygous, it's such a pain. Heterozygous for 1 gene. And that's important because that underpins the math. If that assumption isn't true, then, it's just not going to work. 2, if you have 2 or more different genes, the alleles for 1 gene do not affect the alleles for another gene. So essentially genes are, at least under this assumption, acting independently. Right, that's actually called the law of independent assortment. And again, that's actually one of the more useful assumptions that we can test. And we'll actually be doing that in lab with the fly cross, right? So really the fly cross that we're going to be doing over a number of weeks is testing that assumption, whether it holds true or not. And then we've got some generic ones, right? reproduction is random. And so that's both mating and fertilization. And you'll probably, so as we go through these, you'll be thinking like, yeah, that doesn't like sound right. You know, reproduction isn't random. I mean, I reproduced with my wife for a particularly deliberate reason, right? It wasn't I just met her in the street and there, you know, there you go, now we have kids, right? So clearly this is not a real world assumption, right? These are all part of the thought exercise. 4, no mutations. In other words, no new alleles. And whenever I type that, I always think back to the no capes scene from The Incredibles for some reason, which is one of my favorites. And then the last one, and you can probably come up with others, right? This isn't an exhaustive list, is no selection for, you know, a particular genotype. So for example, if you, if the homozygous recessive genotype resulted in that individual, that fertilized zygote dying, then that would completely skew the results you get from that Mendelian cross, right? So these are all very much part of the thought exercises, right? So these all hold true for, oh, don't mind me, monohybrid and dihybrid crosses. They do not hold true for real world crosses necessarily, but they're a thing that we can test our, you know, we can collect our data and test to see whether our assumptions hold true. And if they do not, that tells us something interesting about what's going on, genetically speaking. Right, that's really the power of these. These aren't prescriptive, right? These don't say this is how it has to be. This is a way of thinking about what we're going to be doing. Pardon me. And seeing where we are wrong. Right, so that's really the kind of the power of them. Okay, anybody got any questions so far? Forgot to ask this at the beginning about either today's or what we covered Tuesday. It's all fairly straightforward, kind of rather dull stuff, but it's important, so that's why we're doing it. You make a slide about dihybrid. For sure I can. Anything in particular on that slide that you want going through? Oh, and also another, okay. And this isn't like a hard and fast because the law of segregation applies to all Mendelian crosses, however many traits you're looking at. But by and large, what we're looking at in the monohybrid cross is law of segregation. And dihybrid cross illustrates, there you go, that's a better way of putting it. And that's all about law of independent assortment. So that's another way of looking at things.
I had a quick question. So when I think of like hybrid, I think of like the example of like a red flower and a white flower, and then they make a pink flower together. What would be like a real world example of like monohybrid or dihybrid?
Woohoo. Well, first off, I'm sorry, I couldn't see who was chatting. That's why I'm not addressing you by name. But um, that's actually a really good example of non-Mendelian inheritance. And so that's an example, or one of the few examples of incomplete dominance, right? Where you get like a blended phenotype between that of the two parents. So that's actually kind of interesting. We'll be coming to that later when we get into the whole non-Mendelian inheritance thing. A good example for real life stuff is typically with humans, at least, it's inherited diseases. Really, that's because almost all human traits that are not disease related are multi-gene traits, like hair color or eye color, for example. There's at least, actually, I haven't actually looked at it for a while, but like 5 or 7 genes involved in what eye color you have. Right, so it's not a simple trait. So something like, for example, Tay-Sachs disease or cystic fibrosis. Actually, even sickle cell disease is actually a poor example for other reasons, right? But yeah, say cystic fibrosis, right? It's a mutation in what is it in? It's a sodium ion pump that's expressed in your, the mucosa or the epithelium of your lungs. And so it doesn't allow, basically you pump ions one way and so water flows the other way. Can't remember which direction it is. Right, and so cystic fibrosis, the disease is the recessive phenotype. Right, so essentially if you had two parents who are, one was homozygous recessive and one's homozygous dominant, so normal and disease, and they had offspring, then those offspring would all be carriers, right? They would be heterozygous. Now, if those offspring, which is kind of weird, reproduce with themselves, then you would have your F2 generation of basically a mix of homozygous dominant, heterozygous, and homozygous recessive individuals. Right, so kind of monohybrid and dihybrid crosses aren't something that really you'll come across much in real life situation, because they're very kind of formal, like rigid structure. What they're really, I mean, their real benefit, to be honest, is as something to test something against, right? To test your assumptions against, and also to learn the principles of inheritance too. But the vast majority of interesting genetics is non-Mendelian. I mean, actually, oh, that was Michael. Cool. I said sickle cell disease is not a good example, right? And so sickle cell disease is from a mutated form of hemoglobin, right? And so that results in you, if you're homozygous recessive, having red blood cells which form these kind of sickle shapes, right? Rather than being nice and round, they're like kind of just past a new moon, that kind of like sliver of the moon sort of shape. And they kind of jumble up and they get blocked in the capillaries, and it's really painful, right? And they don't contain as much oxygen. But if you look at it from the perspective of oxygen or the phenotype of sickle cell disease, it's a Mendelian, single gene trait. However, if you look at it from the perspective of resistance to malaria, it's actually a non-Mendelian trait, because if you're homozygous dominant, you're sensitive to malaria. If you're homozygous recessive, you're resistant because the malaria parasite can't get into the red blood cells. If you're heterozygous, you're actually somewhat resistant. Right, so that would be an example of an incomplete dominance. So you see, it's actually kind of depends on what you're looking at as to what it is. And that actually is the reason for sickle cell disease persisting at a higher rate in the population where you have a high instance of malaria. So South America, Africa, and Southeast Asia, I think, you'll find a higher frequency of alleles that that are responsible for sickle cell disease than you will in Northern Europe, for example, or North America. So yeah, cool question. But yeah, again, monohybrid and dihybrid crosses are really kind of thought exercises, right? That's kind of the best way of thinking about them. They don't really, I wouldn't say they don't have any relevance to real world genetics, because as I said, you know, many congenital diseases in humans are single gene dominant recessive deals. So, but yeah, we can kind of get into the weeds there, to be honest, because anytime you start talking about real world examples, there's always caveats and exceptions and weirdness going on. Okay, so I'm going to go back to the beginning now. Mendel, smug looking chap. Wasn't too bad looking, to be honest. You know, if that's your kind of thing. Lowe's a monk, so that didn't do him any good whatsoever. And the main kind of takeaway from his work, right, it's actually kind of funny because it went into obscurity for decades, right? He published this in the 1850s, give or take, or wrote his treatise. Right? So wrote a book on it. And it wasn't really kind of picked up on until the early 1900s by someone called Thomas Hunt Morgan. I don't know what you're doing there. Get out of it. Anyway, the big deal about Mendel is well, two things. One, he really did fabulous work on formalizing the scientific method. Right, and this is something that he isn't often credited with. He had a hypothesis, he designed an experiment, and he collected and recorded data. And then he worked out what those data meant. Kind of up until that point, a lot of science was actually really, biology wasn't even biology then, it was called you were called a natural philosopher. And so you'd kind of go around and go, yes, birds. Okay, I think such and such is happening. And if I shout loud enough, everybody will believe that too. And in some ways, Darwin was actually he was a natural philosopher. He just did a better job of laying out his thoughts and observations, right? But he didn't do experiments. He just observed stuff, right? Mendel actually did experiments. And so that was kind of a cool thing. But also, he, using that kind of formal approach, proved the idea of particular inheritance, right? And so this idea that what was passed down between generations was kind of discrete particles of information. And you didn't know what those were or what form they took, only that they were discrete, right? And they're not discrete as in like they kind of quite wandering around, but they were separate. Because, sorry, gotta show you this. Right. Look at that. My God. Wish I could do that. Yeah, the life. Because before that, there was this kind of big argument between people who thought inheritance was blended versus particular. And so kind of, oh, there she goes, rolls over on the side. Yeah, blended inheritance was this idea that if you had kind of like incomplete dominance, like a tall parent and a short parent, you know, the offspring would be in the middle. Like, well, if you think about it, what that would result in is that you'd essentially get everybody, you know, in terms of height, moving more and more and more to an average. You'd never get outliers, right? So Mendel actually showed that inheritance was particular, right? He used P's, which are singly the most boring genetic model to work with, in my opinion. But he had good reasons for doing so, because I guess he could eat them, right? That's always a plus. And they're pretty easy to work with, you know, and didn't take too long. This funny thing, it says relatively fast generation of 1 year. I'm like, holy crap, that's not fast. You know, do one experiment a year. I'm used to doing one experiment every couple of weeks. Anyway, so I wouldn't have chosen P's. I would have chosen something better. But so essentially, he used P's because they're just easy to work with, right? Collect lots of data, and they had a bunch of discrete phenotypes. And essentially, those are observable ones, right? So there's a lot more to these than kind of first meet the eye if we're looking at molecular level. But at a kind of a gross physiological level, they're pretty straightforward to kind of get at. I actually, when I teach this in person, I use cats. Cats are way more interesting than P's, I think. And they're a lot cuter as well. So I'm not going to go through that because that's not super interesting. But I already covered this. Now we get to a monohybrid cross. Listen, dog. Sorry, I've got to share this space. There you go. Oh, took her out for a walk this morning. It's kind of hot, so she's a bit flaked out. And the other one's just lying on the floor sleeping. Okay, so let's get back to our monohybrid and dihybrid crosses. We'll probably do the dihybrid cross next class, because that takes a while to kind of get through. It's some not tricky stuff, but stuff. But a monohybrid cross is relatively straightforward. And as you can see, it has 3 parts to it. This is always the same. The traits might be different. We might be talking about seed color. We might be talking about fur color, or fur length, or whatever, right? But it is always the same layout. So I can tell you, hey, I got a generation of individuals in a 3-to-1 phenotypic ratio. And you'll go, oh, that's a monohybrid cross. And you're probably going, what the hell is he talking about? Okay, down here is our 3-to-1 ratio of F2 progeny. Go on, Naomi, hold it together. Coffee is coming. It's funny, actually, this drink that I had beforehand, this is a nasty ass like caffeine drink that my kids had for some reason. Don't know why. I was like, I'm not going to make it through genetics if I don't have some caffeine. So yeah, it's like, and I got to tell you, to be completely honest, Mendelian genetics is pretty dull, right? But it is super, super, super important to understanding all the interesting stuff. That's why we learn it. Not because Mendelian genetics is interesting, because I'll be completely honest, it's not, right? And it's not super relevant to real life either, right? There aren't many instances where we can say, ooh, that's a Mendelian trait. But it allows us to actually get into the kind of nitty gritty of how the interesting stuff works. So we have to just like grit our teeth, push through it, and come out smiling on the other side. So these 3 generations are what we talked about kind of at the bottom of the PowerPoint. In the P generation is always 2 true breeding individuals, right? And you don't necessarily know which one is dominant, homozygous dominant, and which one is homozygous recessive yet. So here's a thing. You can actually, and I'll almost certainly quiz you this in the exam. I think there's always at least 1 or 2 questions about this. You can tell what the genotype of the parents are by looking at the progeny. So you cross these 2 homozygous individuals together and you'll get only heterozygous offspring. And basically, the F1 generation, which are hets, and here's my shorthand because typing out heterozygous is miserable if you do it enough times. So I just use hets and homs, homozygous and heterozygous. They always display the dominant phenotype as they have 1 dominant allele. So if you want to know what which parent is homdom, homozygous dominant, look at the F1s and it'll tell you. So in this particular image, which is the dominant phenotype?
Round seeds.
Yeah, exactly. Right, because that's the one that you see in the F2, in the F1s, sorry. So you cross these together, and this is why we don't use human examples, because, you know, brothers and sisters mating is a little bit icky. Well, it's a lot icky, right? So we just don't talk about that. And this isn't the British royal family either, so it doesn't apply. Or actually, yeah, at some point, before the end of the semester, remind me to walk you through the Spanish Habsburgs family tree. You can look it up yourself. It is just like absolutely gross. Anyway, you cross these together, and so you're crossing heterozygotes with heterozygotes, and you'll get F2s that are in a 3-to-1 phenotypic ratio. So the F2s will always display a 3-to-1 dominant to recessive phenotypic ratio. And that's the result, oops. A 1-to-2-to-1 homdom het homozygous recessive genotype ratio. Basically, the homozygous dominant and the heterozygotes get added together. That gets you the 3. Now it's probably, you're probably like, okay, sure. Well, we're just going to go through this because this is good. Oh, you weird dog. She sleeps with her eyes open. Isn't that strange? Let's see if I can get this to reach. There we go. All right, so this is my whiteboard. Da, da, da. There's the camera. Pens. Oh, damn it, what's my wife done with my eraser? She tidies this up at the end of every semester and I have to go scan around looking for things. There we go. Yeah, don't know what that is. All right, so the hell, where's my black pen gone? Okay, so this is a pretty poor kind of version of what I'd normally use in class, but it's better than nothing. And it's way, 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 way easier to yeah, thanks, dog. Appreciate it. Way easier to draw stuff out than it is to describe things with words. You comfortable? Yeah, good. All right. So, and what I'll do is I'll take a picture of this and I'll upload those pictures to Blackboard along with the chat as well. And one thing that I emphasize that you have to do whenever you're working out genetic cross is figure out what alleles you're going to get in the gametes. For a monohybrid cross, this is really easy. For a dihybrid cross, it is not. For a trihybrid, quad hybrid, it gets crazy, right? So it's kind of like what I talk about in lab safety briefing. It's all about good habits, right? And this is a good habit for genetic crosses. And naturally, you'll be doing the same for the fly linkage experiment too. Excuse me. Thank you. So for your P1, you're going to have, or let's say we're going to use letter A. Can you all see that clearly enough? Let me stop the share. Where's the cursor gone? And that will hopefully make it a little bit bigger. Does that help? Yeah, cool. If I forget to do that, just remind me. Sometimes I do. Right, so again, this seems like fairly, redundant almost, but those are going to be the alleles or the gametes that you're going to get from those two individuals. You're only going to get capital A or a dominant allele in gametes because this is homozygous dominant. You only ever get a recessive allele because it's homozygous recessive. There are no other alleles for that gene. There will be for other genes, but we don't care about those because we're not looking at them. Right. This is what we want. I've never actually taught with a sleeping puppy in front of me. It's a little distracting. All right. Actually, I got to take a picture of this for posterity. There you go. Post that up as well. So you just stick these two things together, right? So now we're going to get our F1s. And as you can see, they can only be heterozygous hybrids, essentially. No, that wasn't comfortable. This is ridiculous. That's the latest pose. Yeah, you're a ridiculous dog. All right. I like slapping myself. Focus. Focus on teaching, not on dogs. Now we've got to do the same thing again, right? What gametes can you get from this individual? Question to the class. What gametes can you get from a heterozygote? Stop looking at the puppy, look at the board. All right, I guess you can do both now. What different gametes can you get from, potentially, yes, but we've got to work through it formally though. So if we're heterozygous, we have both a dominant and a recessive. Yeah, there you go. So we're going to have dominant. I'm going to put little arrows there. Actually, probably shouldn't because that's kind of put offspring. What ratio are we going to get these in? if I were to look at 100 gametes, how many of this versus how many of this?
Three to one.
No, that's the F2 generation. Jasmine and Destiny and Mika. Mika or Micah? One of the two. Are correct. It's 50-50. Right? Because this is what the law of segregation is all about. Which allele goes into which gamete is random. Good answer, but it's good to hear that because then I can, help you steer into the right path. 3 to 1 is about the F2 generation, right? So don't get confused with that. This is going to be a 1 to 1 or 50-50. Right, so that's going to be in a 1 to 1 ratio. Dog, your head's in the way. There you go. So if we're going to cross these individuals together, we're going to self-cross them or self-fertilize them, which is a lot nicer in plants than it is in animals. Then we're going to have the same on the other side, right? So this is, I'm just going to get rid of this. Let's put that up here instead. Now this is starting to look familiar, right? This is our Punnett square, right? So essentially this is one F1. This is another F1 parent. It's actually a really cool XKCD comic, which is about what happens when two geneticists go on the first date. Well, they do a Punnett square to figure out what their offspring would look like. Which I always think is kind of funny, kind of nerd joke, I guess. And so if you want to figure out what the offspring are, you just add these together. Much the same as we did for these to get the F1s, right? And so the F2, right, are going to be these individuals here. We're going to have homozygous dominant. We're going to have a heterozygote. We're going to have another heterozygote. Oops. Now, technically, it's the other way around if we're going to be really pedantic, but it doesn't actually matter. Unless we're worried about which one came from which parent. And then the final one is homozygous recessive, right? So that's a 1 to 2 to 1. Sorry, you have to. Genotype ratio. But you have to look at the alleles to get the phenotype ratio, right? So we have dominant allele here, dominant allele here, dominant allele here. That gives us 3. Let's use a different color. To 1 phenotype ratio. Yeah, run out of space. So you see there's quite a lot to that monohybrid cross, even though at surface, at least, it looks fairly simple. This is ridiculous. Everybody should have a dog to pet while studying genetics. Look at those ears. Anyway. This thing here is my ticket to great SRIs, because at the end of the semester, all you're going to remember is how cute my dogs are, not how bad my teaching is. It's actually an evil ploy. Okay, so that's a monohybrid cross. Really straightforward. True breeding, homozygous P1 parents, heterozygous F1, and just saw that, Louise, I'll get there in a second. heterozygous F1 generation. These generate gametes which are either dominant or recessive, and those are crossed together to create the F2 generation, which always has a 3-to-1 phenotype ratio. Question from Louise. So one thing in your question we need to be careful with is dominance refers to alleles, right? So for any particular gene, you can be homozygous dominant, which is this one, where you have only a, homozygous dominant, where you only have dominant alleles, right? Because we have two possible slots, essentially. Both of those are filled by dominant alleles. So if we say it's homozygous dominant, it can only be this. You can also have homozygous recessive, because homozygous is really about having the same allele for both copies, right? So we have two sets of chromosomes. So for every gene, we have two copies. And if those are both the same, have both the same allele, then you're homozygous. Whether it's homozygous dominant or homozygous recessive depends on whether that's dominant or recessive alleles. Heterozygous is always a different allele for each copy. Does that help? Yeah, it's important terminology to get straight because it really helps with understanding what's going on. Cool. How are we doing on time? 12 minutes left. That's not too bad. Make sure I get there. Ah, man, this camera's such a turd. This camera actually has a lot of history. It's actually a camera, look. It's not a smartphone, kind of crazy. Bought this when we lived in Australia in 2008. So it's actually 13 years old now, which is a little crazy. And we bought it because our previous digital camera, we went to a friend's kid's birthday party, and it was so hot that the chip, the CCD chip that records the image, got damaged. So after that, every picture was pink, which I thought was kind of cute. So anyway, obviously we didn't want to have pink pictures for everything, and so we had to get a new camera. Isn't that crazy? It got that, I can't remember what the temperature was in Fahrenheit. I think it was around 115, 120, something like that. It was really, really hot for an outside birthday party. I don't know what they were thinking about, but anyway. There you go. Cool. All right. I've got this on like a little, one of those dinner tables that you can put in front of your couch. My impromptu office. Anybody got any other questions about monohybrid crosses? Yeah, cool. So one thing that this can actually tell you, if you actually, where is that? Oh, that's not in this one. Oh, actually, it is here. It's kind of useful. So one thing this can tell you is whether or not the gene which you're studying is autosomal or sex-linked. We'll get to sex-linked later in the non-Mendelian inheritance part of the course. But essentially, if this is a autosomal gene, right? So something that is present on your non-sex chromosomes, which for us are chromosomes 1 to 22. It doesn't matter which way round you do the cross, right? Essentially, it doesn't matter whether this individual is the male and this is a female, or this is a female and this is the male. You will always get the same outcome. This is called doing a reciprocal cross, right? If that, and we'll get into better examples later, if that gene is on a sex chromosome, you will not get the same outcome, because sex-linked inheritance works differently, right? Because of essentially only having one copy of the sex chromosome in one of the sexes. Okay, so there's kind of definition of law of segregation, and it's really about the randomness of which allele goes where. And actually, that's something that I kind of forgot to talk about, right? Which is always important when we talk about reproduction. Sexual reproduction, its main purpose is randomness, right? So sexual reproduction comes with a cost. Actually, this is a fun thing to ask. What costs are associated with sexual reproduction as opposed to asexual reproduction? Can you think of what costs could be associated with sexual reproduction? This is a fun thing to think about in terms of, you know, big picture stuff. So when you reproduce, oh, that's a benefit, Lindsay. That's a benefit without a doubt. Although we'll get to biodiversity means something a little different what I think you mean. Diversity in your offspring, without a shadow of a doubt, is a benefit. What's the cost though? So how much of you goes into your offspring in sexual reproduction? Is it 100%? Yeah, it's half, pretty much exactly half, to be honest. You know, if we, well, it is half if we ignore mitochondrial DNA. Right, correct, Mika. So it's half. So you're passing on only half of your genetic information, right? Which, you know, I want the whole cake. I don't want half of the cake. So that's a cost, right? You only pass on half. What else? I mean, is, think about, I don't know, snow leopards or spiders. Does sexual reproduction come, is it risk-free? No. Now, if you're a black widow male, it's like, yeah, man, I wouldn't even want to think about that, to be honest. Right? It's not even free for humans, right? I'm not going to get in there because that's a whole bunch of like sociology stuff, but sexual reproduction comes with a cost. So there has to be a benefit, because this is for higher organisms, like higher than insects or, you know, various invertebrates. This is the way in which we reproduce, right? So there's got to be a benefit, a really big benefit to this. Yeah, there's a big cost, and so there's got to be a big benefit, because that benefit has to outweigh the cost, right? Yeah, it's like you go to university because getting a degree, you know, is a benefit that outweighs the cost of getting it, one would hope. So the benefit is what Lindsay alluded to. Was it Lindsay? Yeah, Lindsay alluded to earlier, is diversity, right? It generates a diversity of offspring or potential offspring, right? And the benefit to that is that the environment is not static. We know that now, like all the news that's going on, and that's been the same throughout history. Obviously, the causes are different. We're almost certainly the cause of this lot, volcanism, or you know, all kinds of different things. Sun cycle is the cause of changes in the environment in our evolutionary history. So environments change, either a macro level, like global, or, you know, continent wide, or even like a micro level in like the bush outside my window, for example, right? And so if you're reproducing, you want to generate offspring which are fit for the environment that you exist in, but also are fit in other potential environments that may change into. So that if things go belly up and the environment changes, say, you know, I don't know, a drought rages through, or one of your food sources dies off for some reason or other, then some of your offspring will survive, right? And so in order to get that diversity, you need randomness, right? And so sexual reproduction all the way through it is geared towards randomness. And that's really one of the underpinnings of the law of segregation. It's random, right? Same with law of independent assortment too. Actually, the same with a whole bunch of different aspects of, and we'll get to this when we talk about meiosis during the cell division part of the course. Right, so keep that in your mind. Like whenever you think about sexual reproduction in terms of monohybrid, dihybrid, or non-Mendelian inheritance, or whatever, it's really all about randomness, because that randomness is a benefit. Right. Obviously, it's hard to see within our own kind of context, right? Because we typically see the downsides of that randomness in terms of kids having diseases and stuff. But for every kid that may have a disease, you're going to have others which have, at least in prior environments, a fitness advantage. Okay, so I'm just going to quickly introduce this, but we'll actually work through it in class on Tuesday. And so earlier I said you can go from genotype to phenotype. You can't always go from phenotype back to genotype. And the reason is the dominant allele. So, here's a question. What's the, and it's sort of on the page in front of you, what's the only phenotype for which you can know without doubt what the genotype is? The only phenotype that you can say confidently, this is the genotype.
Homozygous dominant?
No, because that's a genotype. So what would the phenotype of a homozygous dominant be? It'd be dominant, right? What would the phenotype of a heterozygous individual be? Sorry, stroking puppy. It'd be dominant too. So you can't tell. So the only one left is a recessive phenotype. That one, oh, just getting belly rub. That one has homozygous recessive genotype, always. So if you have a dominant individual, how do you figure out what the genotype is of that individual? Well, the way you do that is by carrying out a test cross. And essentially you look at what comes out of that cross. Is that so? Extra cuteness. Ta-da! Your best pup ever for teaching, you are, dog. Right, so we're going to go through the mechanics of that next class. It's relatively straightforward, but it's actually kind of neat when you think about like what's going on, how you can use it. But it's going to require some board time, and so there's not really time to do that just now. We're pretty much finished up. So have a look ahead. Check out the test cross, because for me, it's really straightforward, right? Because it just, I can speak the language, essentially. For you, it may not be, and that's fine, because that's why we're going to go through it. And also go through the dihybrid cross. That is one that students kind of stumble on a little bit, because essentially we're doubling the number of genes, we're doubling the number of alleles, and we're quadrupling the number of offspring, right? Or different offspring in the F2 generation. Right. Cool. All right, that was fun, everybody. Thanks for staying with me through some of the duller bits, and I have a great weekend. I'll see you on Tuesday. If you have any questions about this, bring it with you on Tuesday, or you can always just email me too. All right. Thanks very much, everybody. Be free, enjoy your coffee or whatever, just staring at a wall if necessary.